Myself, Dr. Noble Kurian, Assistant Professor at School of Life Sciences. My research area is on microbial melanin and its application. Basically, I'm working on bacterial melanin and its application, various applications. Saying more about myself, I done my PhD at Cochin University of Science and Technology, Kerala, and I have about 7.5 years of teaching and research experience from various universities and research institute of India, and I have published about 24 research publication in various reputed journals, and my research interests are on melanin biology, reproductive pathogenesis, and mainly lignocellulosic biomass based bioethanol production. That is my area of research. Coming to my main area of research, which I have been doing since last decade, is on bacterial melanin production. More we are thinking about melanin. Melanin is a black to brown pigment which is widely seen in most of the animals, plants, humans, microbes. It is ubiquitously distributed throughout every organisms. Mainly in the case of animals, or in the case of humans also, it determines the skin color. Mainly skin color is determined by the various concentration of melanin in our skin. Apart from that, it mainly protects us from UV irradiations, and the decrease in melanin can adversely affect the human beings. It can results in more chances for getting cancer, etc. Melanin mainly protect us from getting mutations, UV mediated mutations, etc. It is a UV protector. In the case of plants, it acts as a cell wall strengthener, as well as in plants it was found to be produced as a result of stress conditions. Mainly some of our work which was found that in MS media containing salt, which we try to produce the what is try to grow the plants in salt stress media. It was found to produce this type of melanin into the media. It was also found. Plants, it's mainly plants is mainly help in protecting the cells, as well as it produced during the stress conditions. Microbes also somewhat same, only to produce during the stress conditions, mainly the stress prone areas, such as what are the sea water, sea sediments, deep sea sediments. One of our work found out that the deep sea sediment bacteria produce very good amount of melanin, and desert bacteria, many what heat sensitive bacteria. This bacteria which grows in very hot conditions are found to be produce very good amount of melanin, which one of our studies has been found. And We can see in this picture these bacteria grown inside of a building. Mainly this building is at Chernobyl, which where the nuclear reactor leakage occurred. Due to the area where nuclear reactor leakage occurs, also we can see the growth of fungus, which is producing very good amount of melanin. It was found that this fungus can survive the nuclear radiation. It can absorb the nuclear radiation, survive there, and this fungus is widely studied for their melanin production and various activities. Still, till now, it is being explored. This fungus, special fungus, is being explored so far, and then. Coming to melanin productions in different organisms, we can see different types of melanin is a black to brown color when we generally speaking, but melanins are of different types based on the precursors. We can see most melanin produced via tyrosine, the amino acid, via dopa. From dopa, it can be converted to dopaquinone, dopachrome, and then the monomers of melanin, that is a DHA and DHCA. Usually, it was believed that, or hypothesis that is melanin produced by the bacteria is secreted in the form of this monomers DHA and DHCA, which secreted to the media, the culture media, where it get auto polymerized to form the U melanin pigment. And one of other melanin melanin which is usually seen in humans is a pheomelanin, usually seen in lips and nipples, etc. These cysteine contained melanin can be seen, and these is of red in color. Usually, this melanin is widely seen in the case of Caucasian people also. And one of the widely seen melanin in bacteria is the pyomelanin, usually Pseudomonas, Pseudo-Ultramonas type of bacteria mainly produce pyomelanin. In bacteria, the studies found that mainly the main two types of melanin produced by bacteria is eumelanin as well as pyomelanin. So this pyomelanin is produced via homogenic via tyrosine is precursor 4-hydroxyphenyl pyruvate dioxinase enzyme catalyze the production of homogenisate. By homogenisate, the pyomelanin is produced, and pyomelanin is one of the predominant melanin in bacteria. While coming to fungus, another melanin is there. The precursor is malonyl CoA, that is called DHN melanin. The melanin produced via malonyl CoA, that is a DHN melanin, so the predominant melanin seen in the face of fungus. While in the case of plants, catechol melanin is a melanin which is produced via catechol or benzoquinine and catechol dimers, and catechol melanin is the widely seen melanin in the case of plants. While in our brain, there is a special melanin called neuromelanin also exists, which is their down regulation is associated with usually neurodegenerative diseases. And this melanin these are the melanin is formed by the combination of these catecholamines such as dopamine and norepinephrine and epinephrine forms this neuromelanin pigment. Exact function is not yet known, but still found to be associated with very neurodegenerative diseases. These are some of the major melanin. There are many others minor melanins are there also there. Major melanin seen in different types of organisms. So coming to bacterial melanin, why I am focusing mainly now on bacterial melanin is that first we can see the properties of bacterial melanin. Bacterial melanin, one of the main properties of bacterial melanin is bacterial melanin the bacteria which is producing this melanin can or found to show the resistant to antimicrobial drugs, such as multi-drug resistance. It was shown, one of our studies also shown that multiple antibiotics it is shown to be resistant to. So multi-drug resistance, this type of melanin producing bacteria is been showing it add to its pathogenicity or virulence, we can say. So and this melanin producing bacteria can also shows metal chelating activity, such as it can chelate or it can absorb radioactive waste such as uranium, and it can absorb many other metals, many heavy metals. Many our studies found that it was found to chelate the iron. So this type of many metals it was found to be having chelating activity against the metals. And one of the major advantage why we use bacteria for the melanin production is that bacteria melanin is more purer, more the purification process of bacterial melanin is very easy because the bacteria secretes the melanin extracellularly, while in the case of plants and animals they are bounded to many other chemicals such as proteins. In the case of animals, when we are isolating from plants such as seeds, etc., many impurities can come, and purifying this melanin from these impurities is difficult comparative to bacterial melanin. As bacterial melanin can be secreted extracellularly, it is very easy to purify. But one of the drawback is that bacteria produce less melanin compared to the sources. So we are trying to explore, or we are trying to isolate more melanin producing or better melanin producing bacteria. And many bacteria such as Streptomyces was already found to produce very good amount of melanin, 21.13 gram per liter. And many other bacteria is found to produce moderately good amount of melanin. We are trying to explore the melanin produce better melanin producing bacteria. It's one of our aim in this melanin research. And so we our research was found my research mainly found out a pseudo ultramonas. This is one of the major better producer which I have found out, which was found to produce 1297 microgram per ml of melanin after optimizations, one factor at a time optimization. We can see we use a melanin production media, YMM media, that is which is minimum essential media, where usually we provide tyrosine as a sole source of carbon. The medium appear white during inoculation, and after about 6 to 8 days it will produce a very good amount of melanin where media turns black in color. So melanin products, better producers of melanin we are still searching on and trying to find out. And coming to the melanin's function, mainly because of microbes that help in photoprotection, mainly UV protection. Our research also found out that melanized bacteria can be protected from UV treatment compared to non-melanized bacteria. So thermoregulation is another one, and it can access free radical sinks. It can absorb the what are the free radicals, or the reactive oxygen species produced by our immune cells. It can scavenge this so that it can escape from the our immune system. That is one of the additive factor in the virulence for the bacteria. It can help in nitrogen fixation. It can access electron acceptors. A lot of biological function this melanin have, and these functions can be utilized in various applications, such as it can be used for as a UV absorbent. It's in optical lenses. It can be used as a UV absorbent. That's a sun protection factor. It can provide a very good sun protection factor to the cosmetics such as sunscreens, etc. And usually in the case of vitiligo patients, vitiligo is a disease, it's an autoimmune disease which is marked by the destruction of these melanocytes, so that white patches appear on the body. And these white patches, if it is present in the face, etc., the patient will suffer more than physical problems, or more than what physical symptoms, psychological problems can arise for facing the world, etc. So vitiligo masking creams have been developing so far by using the melanin, as well as melanin has shown very good properties to make it as a good anodic material in batteries such as lithium ion batteries, sodium ion batteries, etc. And it can be used in solar cells for sequestering these sun radiation, solar radiation. It has anti-venom activities. It has shown very good antiviral activities even for against HIV, etc. So various activities it has been shown. It has shown to bound to heavy metals, which can help in water purification systems. So a lot of applications this has been shown so far. So I mean, my studies, it was found out that the melanin secreted by the bacteria, usually some of the melanin, it was found in the in the bacteria cell itself. Usually we are not going to disrupt the cells. Usually we extract the extracellular melanin only. Anyway, inside the cell also, with the time image shown very melanin deposition, but we can see um, the melanin particles which are secreted, showing particles which are secreted is showing very small in size. It's nano, ultra small particles. It's around less than 10 nanoparticle particles also found. Very small particles of melanin has been secreted to the extracellular medium, which we have found out that. And these particles are having a very good stability throughout around 30 days. It has very good, shown very good stability also. So so studies show that the melanin nanoparticles have high stability, and it is a very stable molecule. It can be used as a drug delivery system also. We are working on that also. And this is one of the first reports which we have reported that melanin can inhibit the inflammatory enzymes such as cyclooxygenase, lipoxygenase, myeloperoxidase, etc. So it has shown very good anti-inflammatory activity, and later many other groups has confirmed this in vivo conditions also. It was found to be very good anti-inflammatory activity. One of the melanins, another activity is its anti-cancer activity is it was found to found to kill the cancerous cells, lung carcinoma cells. It was found to kill lung carcinoma cells in a dose dependent manner. And it was found that the inhibition of growth of cancer cell was via apoptotic pathway or apoptotic mechanism. So it can induce apoptosis in the cancerous cells, which can lead to the cell death. That is what we have found out. And further, it was many groups have reported in viva conditions also melanin can be effective anti-cancer agent. More study are needed to make it into a effective drug against the cancer treatment of cancer. So we are still working on that also. Another thing is another application or another thing which we have found out that melanin has a very multiple studies of us. We have found out that melanin has a very good antimicrobial activity. It can inhibit many pathogenic bacteria, many pathogenic bacteria. It can inhibit. The antimicrobial activity of melanin is well known so far. Many reports are there. It was shown that it has very good antimicrobial activity, and we also found out that melanin has shown very good anti-biofilm activity also. It can inhibit the biofilm formed by bacillus as well as pseudomonas brevibacterium, etc. Many biofilm formed by many bacteria has been inhibited by the melanin. And this can be an very good results can be used in very good applications such as coating catheters, surgical catheters with this type of melanin. Mostly these surgical instruments have been this infection due to during surgery occurs due to surgical instrument contamination only. Usually microbial biofilms can stick into these surgical instruments. In order to prevent that, we were trying to produce some coating with some polymer melanin blend so that the biofilm, its anti-biofilm formation activity can inhibit the formation of biofilm in the catheters. So like that, various applications can be done. Various this type of applications can be done by using this melanin pigment. And we recently has found out that during the COVID times, we have found out that melanin precursors such as DHAC as well as L-DOPA found to inhibit or it can it found to bind the spike protein of this coronavirus, and it was found to inhibit the spike protein from binding to the lung epithelial cells. So mainly this found to bound to the spike protein, and earlier reports also found out that melanin has inhibited many of the viruses such as HIV, and many viruses it has found to be having inhibitory activity. We need to work on in laboratory condition to prove more on this activity of melanin against this coronavirus. And recently at Crescent we are working on stress induced or stress the bacteria which is grown in stress conditions such as in the case of salt pan soil. We have isolated salt pan soil from Maracanum salt pan in Chennai. And we have isolated the bacteria which survived in salt pans in salt stress conditions. We are using that microbes in order to screen for melanin producers, and we are getting a very good result. Very better melanin producers we are isolated from the Maracanum salt pan soil, and we are trying to characterize the pigment and characterize the bacteria, and go for further activity studies and further studies on this metabolism studies, all these studies on the melanin production of these bacteria. And so this is all about our research on melanin, and how it is going to impact the the world, which is so in India. Our melanin research has a very great role, because we have a lot of industries which are polluting our waters with waters with heavy metals, contaminants. This melanin was found to be very good. It can absorb most of these impurities, which is impurities which are present. So it can be used in wastewater treatment. And in our country, mainly the areas such as Gujarat, all areas, we can see a lot of vitiligo patients. So in order to treat them, the vitiligo masking creams, all this is a wide, is also of a great use. And we have a great biodiversity, and we have a great type of stress environments in our country, and we can get a good melanin, better melanin producers on different areas of our country. So we need to explore on that. And many other applications we can work on. And I am hoping that I can also contribute to this with other people who are working currently in India on microbial melanin. This is all about my research. Thank you.